So good day to everyone. So I'm Dr. Anup Ashoak and from the Department of Chemical Engineering at B.V. Raju Institute of Technology, Narsapur. So the course that I'll be teaching in today's class would be from Bioprocess Engineering, and the topic that I would be focusing on is the transport across the cell membrane. So coming to an introduction of the transport across cell membranes. So cell membranes or microorganisms or any life as such is basically maintained by cells. So cells have different layers. So that layers that we focus on is cell membrane or the plasma membrane. So for the transport of the different processes across the cells, everything has to be managed by the process of transport that is taking place across these cell membranes. So controlled transport of ions and molecules between the cell and its environment is what is basically the essential to any normal cell functions. So if you see from this image that we have here, you can see that across the plasma membrane, there are different sets of molecules that are passing. So macromolecules can pass through, non-charged molecules can pass through. Similarly, water molecules can go across and also the charged molecules. So the speciality of the plasma membrane is that it can be acting as a semi-permeable membrane. That is, it will allow only certain type of particles or molecules to pass across the membrane layer, whereas it will restrict the flow of certain types of molecules. So coming to the main introduction, so cell membrane is the main barrier which helps in the selective permeability. So what we mean by permeability is the movement of any particular component across the different sections. So this movement might take place due to different differences. So the main difference that we will be talking about is the concentration difference of these particular components. So the flow of these particles across the cell membrane that takes place helps in the regulation of the concentration of these individual particles that is present within the cell and its environment. So permeation is a kind of a diffusion process that takes place across a barrier of a substance in any particular solutions. So the rate at which any biologically important molecules that will cross the cell molecule or the cell membrane through permeation will vary based on the size of the membrane or based on the charge of the membrane or based on the charge of the particles. So based on different aspects like the size, shape and the charge of the molecules and the membrane, the flow across the particular cell membrane will be identified by the cell organelles. So the process will help in maintaining the composition of the system as well as the pH of the system, let it be the cell system or the environmental system. This can be balanced with the help of the transport that is taking place across the membrane. So coming to the different forms of transports that are there. So primarily the transport process is divided into two aspects. So one is your passive transport and the other one is your active transport. So passive transport, as the name suggests, is where the molecules will flow across the membrane or flow through the membrane without any additional benefits or additional energy requirements. So passive transport can be further subdivided as passive diffusion as well as facilitated diffusion, and active transport can be further subdivided as primary active transport and then secondary active transport. So to characterize the transport process for any given membrane, let it be your simple membrane like the cell membranes, or if it is a plant-based membrane, or if it is an animal-based membrane. So for any of the transport process, to characterize the membrane's transport process, we will be able to devise an equation, and that equation basically assesses the membrane's permeability. So the membrane's permeability is basically given by K, as you can see here. So K is nothing but V by AT into ln or ln of Ce minus Ci naught by Ce minus Ci at a time t. So here V, the capital V that is being represented, is nothing but the cell volume of the cell that we are talking about. The A is nothing but the cell's external surface area. So surface area where the primary material is coming in contact with the cell. So that particular surface area. Then we have Ce, which is nothing but the external cell concentration. So our assumption here is that whatever molecules are present in the solution or in the environment outside the cell, that particular material is flowing from the outside to the inside of the cell material. So based on that, we will take Ce as the external concentration, that is the concentration in the outside of the cell structure. Similarly, we have Ci0 or Ci0, which is nothing but the initial interior substrate concentration. So the initial concentration of a particular substrate material within the cell, that is what is Ci naught. Then we have Ci of T, which is nothing but the interior substrate concentration after a particular time period, which we call it as T. So our calculation is based on how much amount of time is given for the diffusion process or for the transport to take place across the particular cell membrane. So the units of permeability, that is K, is equivalent to that of the velocity. So it can be represented as simply as centimeter by second. So based on the size of the molecule or based on the size of the cells, we are basically limiting it as centimeter per second. Now coming to the passive transport phenomenon. So as I have told you, passive transport basically is divided into passive diffusion as well as facilitated diffusion. So to give you an idea of passive diffusion, so passive diffusion is a type of diffusion which is basically characterized by the movement of substance in the direction of concentration gradient without any energy input. So it is much similar to a simple diffusion process that is taking place. So in simple diffusion process, the main idea is because of the difference in concentration across any particular layer or section, the particles will flow from the high concentration region to the low concentration region. So similar is the case with passive diffusion. So if your plasma membrane is considered as the layer across which the cell has or the particles have to flow, then the particles will flow from a region of high concentration to a region of low concentration. So that depends on the type of molecule that is required or the type of the cell that is required. So based on that, the flow of particle will take place. So the passive diffusion process is basically a spontaneous process. So basically it is dependent on the concentration gradient. So we will consider it as a spontaneous process, and the free energy change that is associated with any spontaneous process is basically given as delta G naught is equal to RT ln of C1 by C2. So the value of delta G naught, that is the change in the free energy change of the system, is basically negative in passive diffusion because the value of C1 is less than the value of C2. So to consider here, C1 is nothing but the initial concentration of the particle or molecule inside the cell as against C2, which is the concentration of the particle or the cell or the molecule inside the cell after the process has completed. So for example, if the process is such that the particles move from the external of the cell to the interior of the cell, so then the concentration within the cell will increase after a particular time period. So that is the reason that we have C1, which is the initial concentration, that is less than that of C2, which is the final concentration. So the penetration of any different substrate material. So these substrate materials have different size and shapes, and because of which the penetration will also be different for different types of substrate materials. So that is what we mentioned here, that it is mainly because of the structure of the membrane. So the structure of the membrane, for example, if it is a porous membrane, and let's say that the pores in that particular membrane are of the size of 50 nanometers. So it will allow any particles which are less than 50 nanometers to easily pass through it. Any size for a size of particle greater than 50 nanometer, the diffusion might not be possible easily, and for a particle of size equal to 50 nanometers, the diffusion will occur, but comparatively some more investment in the process is required. So based on the size and structure of the membrane as well as the individual particle materials that are there, the passive diffusion process is going to vary. So the cell membrane, if you look at the cell membrane, so the cell membrane are basically made of mostly lipid bilayers. So lipids are proteinaceous material, and since they are proteinaceous material, they are long chain amino acids that are present, and because they are long chain amino acids and polypeptides that are present, the flow across the cell membrane that is there will be managed by these amino acid molecules that are present in the lipid bilayers. So yeah, so the moreover, the solubility of the individual components or individual particles into the lipid bilayers, whatever is there, that also handles how much amount of diffusion is taking place across the cell membrane. So the charged molecules and polar substances have small lipid solubility. So if you look at the charged molecules or any polar substances like Na plus, potassium plus, calcium plus or calcium 2 plus, so they do not pass through the cell membranes by passive diffusion. So basically for these particles or the charged ions or molecules or polar substance to pass across the membrane, they require some additional forces, and that is what we'll be discussing as we go into the next slides. So the next part of the passive transport is nothing but facilitated diffusion. So facilitated diffusion is also called as carrier mediated osmosis. So why it is called so, we'll discuss. So it is basically nothing but the facilitated diffusion is the movement of molecules across the cell membrane via a special transport protein that are embedded into the plasma membrane by actively taking up or excluding ions. So in case of the facilitated diffusion, if you see, an additional component layer is present within the system. So this additional component layer that is coming in contact with the cell membrane or the plasma membrane that we have, this is basically a carrier protein. So why this is being attached is that, so there are certain kinds of particles which may not easily diffuse through the cell membrane. So when you are attaching this carrier molecule, so this carrier molecule will provide the pathway for these molecules or ions to pass through the cell membrane without any difficulty. So in case of the facilitated diffusion, if you increase the concentration of the solution or the solute outside the cell membrane, then what happens? This will increase the concentration of the solute, thereby increasing the concentration gradient. So since the concentration gradient is increasing, the transport process will also increase. But the major problem with this is that after a particular level has been reached, which we call it as the saturation level. So once the saturation level has been reached, then further no more changes can take place across the plasma membrane, even if the concentration is going to increase over the period of time. So that is what we have here. So in this particular type of facilitated diffusion, what is basically happening is that only specific components are transported. So not all components will be transported across this carrier protein. So carrier protein is also specific in nature. It will allow only certain types of molecules or certain specific type of molecules to pass through the cell membrane. So the idea here is to identify specific type of molecules which the carrier protein will support, so that the molecules from the external of the system can flow through easily across the cell membrane into the cell concentration. So the carrier molecules that you are basically, as we are telling, it is basically proteins in nature, and these are basically known as permeases. So permeases are a special kind of protein that help in the permeation process. So permeation is nothing but your diffusion process, and these carrier molecules or carrier proteins are called as the permeases. So an example of the facilitated diffusion is when glucose is being absorbed across the cells through the glucose transport 2 in the human body. So the glucose transport 2 is an example of the carrier protein that helps in the absorption of glucose across or into the cells of the human body. So that is the idea about diffusion. So coming to the other operations that is possible in any cell kind of a process. So filtration is the next possible process that we'll be discussing about. So in filtration, so I think everybody is clear with the general idea of filtration. So filtration is nothing but it is the movement of water and solute molecules across the cell membrane due to the hydrostatic pressure generated by the cardiovascular system. So here we are specifically focusing on the filtration that is taking place in the human body or in a live cell that is there. So in this case, what is basically happening is that any kind of molecules, so these small molecules that you see in yellow here, so that can pass through this filter membrane or the membrane that is present in the section. So that can easily permeate through or diffuse through the filter media that is present, whereas these large sized red molecules that you see, these cannot pass through easily through the system. So because of which you have a retentate section where the red molecules and along with it certain kinds of yellow molecules will also be collected, whereas into the permeate section you will have a only dispersion of the yellow kind of molecule, which helps or which is the smaller in size compared to that of the filter media that is present. So the process of filtration is basically governed by the size of the membrane pores. So for example, if you see here, the size of this particular membrane is easier or easy for the transport of this particular yellow kind of molecule. So the yellow molecule will easily pass through these membranes, whereas the red molecules will not diffuse through the or filter through this particular membrane material that we have attached. So an example of the filtration process is the Bowman's capsule. So Bowman's capsule that is present in the kidney are very small in size, and only albumins, the smallest of the proteins, have the chance of being filtered through. So the filtration process that happens in our kidneys, So that is basically and the best example of the filtration process. So the filtration process, the membrane in our kidneys is basically known as the Bowman's capsule. So the next process that we have is basically called as osmosis. So I think everybody knows what is reverse osmosis, because that is how we get the pure drinking water in most of the cases. So here osmosis is nothing but the net movement of water molecules through a partially permeable membrane from a solution of high water potential to an area of low water potential. So the difference here from the usual cases is that here also the flow is taking place from high concentration region to low concentration region. But here the concentration that we are going to consider is the concentration of water and not that of the solute. So in case of osmosis, the water will flow from the high concentration region to the low concentration region. So if you look at the image that we have here, so there are basically three different types of osmosis process. So one is when the system is isotonic solution, the other one is when it is a hypotonic solution, and finally we have the hypertonic solution. So in case of a hypertonic solution, if you see from this image here, you can see that the cells have shrinked in size. That is basically because the concentration of water that is present in the cells is comparatively higher than the concentration of the water that is present in the external surface. That is, in this particular case of hypertonic conditions, the water will flow from inside the cell to the outside of the cell across the cell membrane. So whatever is happening is happening across the cell membrane. So in case of hypertonic solution, what happens is that the water content that is present inside the cell will flow to the outside of the cell across the cell membrane, thereby decreasing the water content in the cell. So that is the reason you see here that in case of a hypertonic situation or a hypertonic solution, the cells have started to shrink down. Next is the case of an isotonic condition. So isotonic condition is a simple case wherein the amount of water that is flowing in and the amount of water that is flowing out across the cell membrane is maintained at the equilibrium condition. That is, the amount of cell or amount of water flowing into the system is equal to the amount of water that is flowing out of the system. So that is basically what we call as the isotonic condition. And finally, coming to the hypotonic condition, in hypotonic condition, what happens is basically that the amount of water that is present in the external of the cell is comparatively much higher than that of the water content that is present inside the cell. So what happens in this case is that due to the high potential difference between the water concentrations, water from the external source will start to pour into the cells, and the cells will reach a particular condition wherein the water becomes so excess that the cells will start to burst out and the water within the cells is completely leaking out. So this particular case where the concentration of water exterior to the cell is comparatively higher than the concentration of the water interior to the cell, and which leads to the flow of water into the cell, and finally the breakage of cell is taking place. In that particular case, we call it as the hypotonic condition. Right, so this was basically about the passive diffusion process. So now coming to the second part of the different process. So we have what we call it as the active transport. So we had the passive transport, which we divided as the passive diffusion and the facilitated diffusion. So now we come to active transport. So to generally introduce what is active transport. So active transport is the movement of molecules across a cell membrane from a region of lower concentration to a region of higher concentration against the concentration gradient. So in case of your passive diffusion, the main idea was that it will flow from a region of higher concentration to a region of lower concentration. But in case of the active transport, if you see, it is flowing from opposite to the original potential gradient or the concentration gradient. That is, in this particular case, whatever the concentration difference is there, that is for the particular movement of the cell, it will flow from a region of lower concentration to a region of higher concentration. And for this particular process, it requires a particular energy, and that energy for the cell across the cell is being supplied by the ATP. So ATP, if everyone remembers, is nothing but adenosine triphosphate. So this is how energy is being stored in case of cells in our body or in any live cell. If you see, cells basically store energy in the form of ATP. So the energy in the form of ATP has to be supplied to the cell membrane so that the molecule will move from a low concentration region to a higher concentration region. right? So this is usually done so that we can accumulate more amount of ions, glucose, or amino acids. For an example, let's say that your body suddenly requires larger amount of glucose content. So in that case, if sufficient amount of glucose is not present within the cell, it will start to pull the excess content from the outside of the cell, right? So the excess content that is present, it will be pulled from the outside of the cell membrane, and then it will be pulled into the cell so that the glucose can undergo whatever reaction it has to be take place, and then the required amount of energy can be obtained from this glucose dispersion process. So coming to active transport is also of two types. So it is of primary active transport that uses ATP, that is energy in the form, and we have secondary active transport that basically uses the electrochemical gradient. So the example of active transport includes the uptake of glucose into the intestine. So as I was telling previously, so the uptake of glucose into the intestines is basically nothing but your active transport process, and the uptake of mineral ions into the root hair of the cells, root of hair cells of plants, that is also an example of your active transport. So in detail, we look into what is the primary active transport. So primary active transport, or also called as the direct active transport, it utilizes the metabolic energy to transport the molecules across the membrane. So as you see, this is your cell membrane, and in this particular case, we are transporting the potassium ion that is there from the exterior of the cell to the interior of cell, and for that purpose, we are utilizing energy in the form of ATP, which is later being converted into ADP. So from the adenosine triphosphate form, it is getting converted to the adenosine diphosphate form along with a phosphate molecule that is getting released. So in this particular case, if you see, we have something called ATPase. So ATPase is a protein kind of molecule, this yellow structure that you see. So it is the protein molecule that is attached to the cell membrane, which helps or aids in the transfer of the cells across, or the individual molecules across the cell membrane. So the most common substances that is being transported in this primary active transport include mostly the ions. So you have sodium ions, potassium ions, magnesium ion, and calcium ions. So these charged particles, usually the ions that we have, or basically the charged particles that we have, they require something called the ion pumps or ion channels, right? So these ion pumps or ion channels are basically these yellow structure that you see across the cell membrane. So in this particular case, where we are transporting sodium or potassium across the cell membrane, ATPase is a protein kind of a molecule which acts as your ion pump across the cell membrane. So most of the enzymes that perform, or most of the protein kind of material that perform this type of transport process, is basically termed as ATPase. So the primary ATPase is universal to all animal life. So this is basically your sodium potassium pump. So whenever there is a decrease in the sodium levels or potassium levels in your body, this particular system is activated, and thereby either the potassium or sodium, the required quantity, is absorbed from the exterior of the cell and brought into the cell with the utilization of energy in the form of ATP. So because of this flow from the low concentration region to the high concentration region, that is why we have a direct connection between these two ions. So in this particular case, we are pulling in the potassium ion from the exterior of the cell to the interior of the cell and replacing the same. So to maintain the neutrality of the charge, what is basically occurring is that the similar quantity of charged particle in the form of sodium plus is being dispersed out into the external of the cell structure, right? So primary active transport uses the redox energy in the mitochondrial electron transport chain. So if you remember, mitochondria is nothing but the powerhouse of the cell. So if you look at the different cell structures that we have, and if you focus specifically on mitochondria, so mitochondria is nothing but your powerhouse of that particular cell. So whatever energy that has to be supplied to this particular system, that is being supplied from the mitochondria within that particular cell. And in cases of plant cells, it will be utilizing the light energy that is being produced from the photosynthesis for the same process to take place. Next, coming to secondary active transport. So secondary active transport is also called as the coupled transport or the co-transport, because it uses the energy to transport molecules across the membrane without directly combining with your ATP molecule or the energy requirement in the form of ATP molecule. So this particular case, the main focus is on the electrochemical potential difference that is being created by pumping ions in and out of the cell. So for example, what we mean by electrochemical potential is that the difference in the concentrations of, or the difference in the charges of concentration across the cell membrane. So because of these differences across the exterior and interior of the cell membrane, the particular ion is being pulled into the system. So here also, if you look at it, you have a yellow kind of a structure which is used, or which is an attachment of protein to the cell membrane. So this particular site or this particular structure that is being attached, it specifically focuses on what type of requirement is there. So in this case, if you see, there is a sodium plus binding site and there is also a glucose binding site. So the sodium plus, that is basic requirement, it is being pulled into the cell membrane through the sodium binding site, and similarly along with this sodium ion, the glucose molecule that is present is also being pulled in it, so as to maintain the electrochemical potential across the two sections of the cell membrane. So usually if you see, the sodium ion is commonly co-transported across the cell membrane, whose electrochemical gradient is then used to power the active transport. So Na plus is being pulled into the system so that electrochemical gradient is being developed, and in order to balance it out, the glucose molecule that is present in the external layer is being pulled along with it, so that the overall electrochemical potential across the cell membrane is thoroughly maintained over the entire section. So the energy that is basically derived from the system across the cell membrane is usually used as the energy for this entire transport process to take place across the secondary active transport. So in special case scenarios, you have something called the co-transporters. So co-transporters are of three different forms. So one is your uniporter, second one is the synporter, and finally you have the antiporter. So what is the benefit of this is that in a uniporter, only one particular type of material can pass through that particular protein molecule that is present. Whereas in case of a synporter, it will allow the transport of two different molecules from any one side to the other side. For example, if it is from the external to the internal, then both the molecules will flow from the external of the cell membrane to the internal of the cell membrane. That would be an example of a synporter. And finally, coming to antiporter, in case of an antiporter, what happens is that when one molecule is pulled from the external to the interior, in a similar scenario, another molecule has to be pulled from the interior to the exterior, so that the overall balance of the system has to be maintained as equal. So uniporter is when only one particular particle or molecule is being pulled. Synporter is when two particles or molecules are pulled together from either external to interior or from interior to the exterior. And antiporter is when the opposite. It is something like equivalent to a co-current kind of situation. That is, one molecule is moving from the exterior to the interior, whereas the other molecule is moving from the interior to the exterior. So this is basically the idea about the different transport process that is taking place in the cell membrane. So we have basically discussed about the primary active transport, and we have also discussed about the secondary active transport. Then we have also discussed about the facilitated diffusion and the simple diffusion process that is taking place. So these are the different methodologies by which the transport process takes place across the cell membrane. So thank you everyone for listening for today's class. I'll come up with more further lectures on different types of process that is taking place in your bioprocess engineering. Thank you.
Like, share and subscribe. Hit the bell icon for more updates.